that the cancer cares only if it also carries the oncogene. And for reasons that are not 100% clear, the presence of that oncogene makes other genes matter. That's the synthetic part of this. And people think that if you could identify those synthetic genes and target them, they'd be very good targets to go after, because only the cells that would carry the oncogene would care about them. So here's a real data slide. And here we're not looking at thousands of different types of cancers, we're just looking at 2 of them, because looking at the data from large numbers is challenging. And so there's 2 types of AMLs here. One that carries a mutation in a gene called FLT3, and another one in a gene called JAK2. And these are very famous genes that are associated with this cancer. And when you're looking at it, every little dot here is one gene. And it's the score that it got by going through this CRISPR method. So on the x-axis is the score for the genes in this type of AML. On the y-axis is the score for this other type of AML. And what you can see is for the vast majority of genes, they're all on the diagonal. That is, the score was the same. This gene did not distinguish whether it was this type of AML or that type of AML. And these are genes that we don't care about, because those are genes that are likely to matter to all cells in the body. So we don't really care about those. The ones we care about are these genes, the ones that only matter, in this case, to this one, or over here, only to that type of AML. These are the ones that are likely to encode these special drivers as well as these synthetic lethal type of genes. And indeed, if we simply look here and say, okay, well, these 2 genes, we know they matter, and indeed they do. One of them lands here. Pretty much it has no effect in this cell line, only in this one, and likewise for that in the opposite situation. So, and one can do this across very large numbers of cancers and very conclusively say that gene is the gene that this cancer cares about, and therefore if we had an inhibitor to that, we should be able to target it. However, you can see that there's other dots here. You can see like around JAK2, for example, there's a whole bunch of other dots. Around FLT3, there's a whole bunch of other ones. And these are the ones that might be kind of these holy grail type genes, that they only matter if you also have this JAK2 mutation. And I'll show you a little bit how we get at those. So this is just 2 lines. Here's another situation where, in this case, this is another type of AML. We actually had no idea what the driver was for that type of AML. So the genome sequence didn't tell us what it was. And so we asked, which gene really matters here, but not in any of the other ones? And sort of mathematically, one can calculate this and then plot these scores. And it turns out that there's one gene that shows up at the top of this list. And this is a very famous gene. This is actually the first oncogene discovered, and this was discovered by Bob Weinberg, who spoke in this forum, I think last year or the year before. This was the first oncogene, and Bob discovered the first oncogene. But it turned out that this gene in this type of cancer didn't have the type of mutation that he had described. So when you looked at it in the genome sequence, it didn't raise any red flags because the mutation was not the kind that he had shown and other people had shown were important in cancer. It did have a mutation, though, but a very odd mutation that then we went on to show is actually also a cancer driver. And so this genetic system pointed us to a classic gene, but it had a different kind of mutation that we didn't recognize, that again, computationally, we never would have picked. So KRAS is the most common oncogene in human cancer, and KRAS, and there's a couple of related genes to KRAS. The problem with KRAS is that it falls into the so-called untargetable class of proteins. That is, chemists can't think of a good way of making an inhibitor to this protein because of the nature of what kind of protein it is. It simply doesn't have a place that you could stick a small molecule into and make a drug. And so people have been very, very interested in finding this other class of interesting genes, these so-called synthetic lethal genes with KRAS. And again, there's large numbers of people trying to find these. And so we asked, could we do that from our data set? And let me show you what that looks like. This is the last data I'll show you. So this illustrates the synthetic lethal concept for the one type of cancer where I think it's been validated, and this is breast cancer, and particularly cancers that have mutations in 2 very famous genes called the BRCA1 and 2 mutations. These are the ones that there was a recent report of a new method using blood samples to identify these, sort of a a more cost-effective method. And this gene normally has a normal role in the cell that when you lose it, makes it sensitive to inhibiting another gene. And that gene is shown here as something called PARP. And don't ignore some of the details on this. And so if the cancer cell has lost either of these two cancer genes, the BRCA1 or 2, and you give this inhibitor of another gene, the cancer cell dies. Normally, inhibiting this gene doesn't matter because we have a backup for it, that these BRCA1 genes do. And so normal cells don't care, but cancer cells do care. And this has actually been followed into the clinic, and there are now clinical trials with these so-called PARP inhibitors that have been showing some success. But this is really the only cancer for which we have this example. So the question is, can one find this type of interaction for other types of cancer, particularly these KRAS cancers, which are the most common types of mutations in cancer? So what I'll show you is I think the answer is yes. This is actually unpublished data, and I'll tell you a funny anecdote about this. So there are actually several types of RAS genes, as I alluded to, and in AML, there's two types. One is called NRAS, and one is called KRAS. And what you're looking at here is the score for the KRAS gene if this cancer cell has a mutation in KRAS or it has a mutation in NRAS. And the only thing you need to know here is that if it has a mutation in KRAS, it really cares about that gene. So this is the classic cancer paradigm. If it has the oncogene in it, if you delete that oncogene, the cancer cell cares. And so if we could make an inhibitor to KRAS, it would be very effective. But again, we can't. The same thing is true if you have a mutation in NRAS. The cell cares about NRAS, and it doesn't care about KRAS. But NRAS is just as bad to target as KRAS. So the question becomes, is there a gene that if you have either of these two mutations, the cell cares about that we could target? And the answer is yes, there is. There's one only, and completely unexpected, and it's called RAF1. So you can see these scores are negative if you have either of these two mutations. And I have to say, this is the somewhat funny anecdote. So this name is actually very similar to another gene that's very famous in the cancer world called BRAF. And I didn't know, and one thing you guys may not know is that there's official gene names and then there's sort of colloquial gene names. So there's like the names that we use as a community, and then there's the official names that are in the databases. And this is one of those official names. And I actually didn't really know what this is, but I assumed it was that other famous gene. So I didn't think that this was that interesting because we had identified another famous cancer gene called BRAF as sort of its common name. But then I was presenting this to a drug company. I was trying to get funds to do more of this research, and their eyes completely widen up. And they said, is that true? And I said, yeah, it's true. It's probably this BRAF thing, which you guys are making inhibitors. And they said, no, it's not BRAF. This is another gene called CRAF, in fact, which no one has made inhibitors to at all. And so now I've actually told this to a couple of companies, inadvertently, really, because I didn't think it mattered so much. And so we're actually now involved in a couple of collaborations to try to make inhibitors to this gene. And so it probably reflected my ignorance of the gene nomenclature, but also the fact that this was unexpected to them. So this was an unbiased approach, and the number one gene is one that, despite the decades of research that have gone into NRAS and KRAS cancer, starting with Bob's work at the Whitehead, did not identify this gene as essential in this type of cancer. So I think it's a very nice example of how if you simply don't have any predictions, you sort of go into a completely unbiased way and you just tell the cancer cell, give me the answer, and then you do that experiment in many, many cells. This is data from only around 14, but eventually if we did this in hundreds or thousands, we could find all of these types of relationships and really identify, I think, a new class of targets to go after in cancer, which I think this is an example of really just the first one. So I think I'll stop here and hand it back to Rick, and if you guys have any questions, I think maybe I'll come after and chat with Rick. So thank you very much.
Thank you, David. I think I'll just add two things. One is that what you've seen is really very special from my point of view, because this type of thing really can't today be done anywhere else the way David can do it. And that it's not just the promise of trying to do it and see what happens. There are real results coming out. And what especially excites me is in the future of personalized medicine, the idea is that we identify the specific gene that is going to become the target in your cancer. But cancers are really quite complex, and as David pointed out, there are redundant pathways. And so finding the essential genes for which there aren't redundancies, like he's just found here, is really a critical goal. And I think this is one of many exciting new results that are going to come out of that screen. So I want to turn now to the second leg on this stool, genome analysis. And I want to focus on what you can do once you have information about these essential genes. The type of things we can do with such a genome analysis is also technologically driven. And we live in a world where the most recent knowledge, the most recent insights get paired up with cutting-edge technologies. And what we're trying to do is to take that information and understand what we call pathways. So none of these genes or their RNA or protein products live alone. They usually live in a pathway or a network of interactions. And mapping those is critically important for us to fully understand the sorts of drinking buddies that go along to affect that pathway. And there are two additional things I want to very briefly tell you about, and that has to do with how that 2 meters of genome exists in a structure within each chromosome. These are insights that we've gained literally in the last 12 months that I'm going to explain to you. And then how the alteration of these pathways and the alteration of chromosome structures work together in the cancer context. One of the things that's been very exciting about this is that this is not something we realized a year ago or 2 years ago or 3 years ago. This is something that has come about very, very recently. It's really what I think of as an inflection point, because the ability to do high throughput genome sequencing has given us some information. The understanding of how genes are controlled has given us some information. And this understanding of the kinds of vulnerabilities, what David is calling essentiality genes, is coming together. And at just one level that I really want you to remember, it has come together in a powerful way just recently. And that is the realization from all of the human genetic studies that have been designed to identify human sequence variation associated with disease. We've come to understand that only about 5% of human genome variation associated with disease is changing protein structures. The other 95% or so are changing the gene control regions in the genome. And marrying up that information, knowing what these gene control regions look like and how they function, together with this understanding of protein variation, I think is the key to cancer's future. So let me tell you a little bit about this inflection point. It is in the early 2000s that the public effort to sequence the genome, led by Eric Lander at that time at Whitehead Institute and doing this draft genome, led to what is really our current understanding of our genome sequence. And it was about 10 years later, only 3 years ago, that a group called ENCODE, funded by the National Institutes of Health, came together to identify what they called the machinery. And this was really the proteinaceous machinery of gene control, and it was the sites on the genome where that proteinaceous material operated to control genes. And this year, what's come apparent is a combination of this information that's led us to an understanding, at least a very early understanding, of what we think of as global gene control. How all the genes in any one cell are controlled in a coordinated type of circuitry. So let me tell you just a little bit more about this. There are new technologies that allow us to use high throughput sequencing in combination with an ability to actually reach in and grab this proteinaceous machinery on the gene control regions to identify what we think of as core regulatory circuitry. Core regulatory circuitry, if you focus on that little diagram on the upper left of brain where there's sort of a square of 3 red rectangles, those represent genes that encode a special kind of protein called transcription factors. These are the gene regulators. The gene regulators then do something that's really pretty remarkable. They regulate their own genes. So the key regulators in the control room of your cells operate on their own genes and form what we call an autoregulatory loop. And that's what's represented for the brain here, on the left for the heart, the left for the pancreas. These are the control centers that then operate on the rest of the approximately 12,000 genes that are on in any one cell. So we're coming to understand what this regulatory circuitry looks like in draft form for every normal human cell. And that's exciting. And it's exciting because if you look at Bob Weinberg's diagram for what the altered pathways are in gene control, they all converge. All of the pathways converge on gene expression. So there is altered gene control. The two big kind of themes in altered gene control are signaling pathways, those pathways by which cells sense their environment and tell their genome what to do about it, and then the circuitry, the gene control circuitry itself. And those two pathways happen to be the places where David Sabatini and I focused our research during our entire careers. Cancer cells are rewiring this, and what we're finding is that many of the key oncogenes are genes that are producing transcription factors that have gone awry. So the most famous of these is something called MYC. MYC is one of these essentiality genes in most cancers, perhaps all cancers. What we're finding when we profile individual cancers of specific types is that we can find not just MYC, but other genes that produce oncogenic transcription factors that are misregulated, that are in the control rooms of those cells, and they're driving what is a normal cell into a different cell identity. The other area where there's been a really profound change in our understanding is in how chromosomes are structured. And for many years, people have been interested in trying to understand how the genome is folded up in a manner that you can still operate on it. If you take your 2 meters of genome and you squeeze it down into 5 microns, you can imagine it may be challenging for the operations that are occurring in that genome, which include repairing the genome if there's a break. They include, if you're going to divide, making another copy of that genome, or the operation that's occurring constantly in all our cells, which is making that RNA product that is the template for the machinery, which are these proteins. And what we've come to realize is that all the important genes, the genes that determine the identity of the cell, the genes that make a difference for whether you're a neuron or a cardiomyocyte, live inside a physical structure that is brought together by what are called genome structuring proteins. And so all the important genes live in a DNA loop. And I hadn't appreciated this until we published it last October. But all of those important genes live in a loop whose integrity is critical to the control of that gene. If you lose that loop, which is brought together by two proteins bound to the genome that dimerize to form the loop, if you lose that integrity, then the control operations inside the loop begin to occur in other regions of the genome. And control operations that are occurring in other regions of the genome now begin to invade those regions. And so that's why we call it an insulated neighborhood. And you have thousands of these folded structures in your chromosomes that are essential for that cell's identity. And what we've discovered is in tumor cells are deleting the segments that are necessary to form the insulated neighborhood, so that what we call a proto-oncogene, a gene that's behaving normally, that is probably silent in those cells, becomes activated. And it becomes activated because now it's lost its insulated structure. And regulatory elements we call enhancers literally loop in now to what was otherwise an insulated silent neighborhood, activate that gene, and that is called an oncogene. So we live with proto-oncogenes. Most of them are in these silent insulated neighborhoods. And this is a whole new mechanism we had not appreciated by which cancer cells generate oncogenic behaviors. They activate an oncogene in many cases by deleting a segment that is part of this integral chromosome structure. So I want to go back now to what David was telling you. If we have information that's quantitative, that tells us what is the essentiality, what is the vulnerability, what is the score for absolutely every gene in any one cancer cell, we can go and figure out how it fits into the signaling pathways, how it fits into the genome control room, and how it fits into the structure of the genome. And I think we can do this in a way that cannot be done anywhere else. So I want to turn to now the last leg of this three-leg stool, chemistry. And what I want to discuss here is the fact that there's been a revolution in drug design that enables us to get into the drug development space, not necessarily to develop a drug that hits the target that we've identified by these genetics and genomics tools, but rather to get something I'd call a pre-drug, a chemical probe that will, I think, cause us to vastly accelerate solutions, because we can provide this to all academics, and we can provide this to pharmaceutical companies, and that will encourage them to move more rapidly to a really clinically valuable drug. So what we're thinking about here is designing chemical probes that interact with the targets we identify through these essentiality screens and through this pathway analysis. And this is very different than the classical view of drug screening, which is you identify a target, you take that target protein, you put it, or a cell